Morning to dear students. I'm Dr. Ravi presenting the second part of biology, the applications, biotechnology, the applications and the content concerned with it. So in the previous video we had studied about the synopsis of the entire content of this biotechnology applications. In the present video we'll be discussing about some of the interesting questions that can be asked for your uh, the NEET examination. So within the NCERT questions, one or two might be outside of NCERT syllabus, but anyway, we'll have a discussion on that. The first clinical gene therapy was done for the treatment of AIDS, cancer, cystic fibrosis, and severe combined immunodeficiency. So cystic fibrosis and SCID, they have done gene therapy on. Using gene therapy, they have treated cystic fibrosis and SCID. So SCID can also be written as ADA deficiency. So the answer for this would be SCID. SCID is severe combined immunodeficiency syndrome or bubble baby syndrome. So in our textbook, they have not used this word, rather they have given it as ADA deficiency, that is ADA deficiency, adenosine deaminase deficiency. The second question is the site of production of ADA in the body is it in the erythrocytes, lymphocytes, bold plasma or osteocytes? The site of production of ADA in the body is lymphocytes. That is the reason in the experiment for treatment of by gene therapy, we extract lymphocytes from the body and are going to introduce the normal ADA gene, CADA gene into the lymphocytes and correct the malfunctioning gene. So this we are aware about it. So the site of production of ADA in the body is lymphocytes. Lymphocytes are part of WBC. So you would be knowing about white blood cells, the A granulocytes. Under the A granulocytes, you have monocytes and lymphocytes. Granulocytes, there are the eosinophils, basophils, and neutrophils. So lymphocytes are the site of production of adenosine deaminase in the body. And lymphocytes, just like the white blood cells, which have a lifespan of nearly 120 days, they also have a lifespan. So that is the reason we have to frequently do this process of introducing the normal ADA gene into the lymphocytes and reintroducing into the affected individual. The source of genes for high vitamin content in golden yellow rice is, so if you have studied about this golden yellow rice, this is outside of syllabus, but they have mentioned in our book only golden rice. So two genes are from daffodils and one gene from Erwinia red overa. Erwinia red overa is a bacteria. So and two genes from daffodils. You might have read this as a poetry of William Wordsworth. The daffodils, not commonly seen in India, but in European countries, especially Britain, you can see a lot of daffodils growing there. So since they are rich in the carotenoids, they utilized Ingopotricus, utilized two genes of daffodils and one gene of from Erwinia red overa. So that is the reason the correct option for this is both A and B. This is outside of syllabus question from NCERT. It is outside of NCERT. So which one is known as superbug? Again, this is outside of NCERT. The answer is Pseudomonas putida. They are used for bioremediation, for treatment of oil spillage, the crude oil because of the accidental spillage from ships carrying the crude oil, or purposeful intentional leakage of crude oil into the oceans. So they can be treated by using this remediation can be done by using this superbug Pseudomonas putida. The first transgenic crop is cotton. BT cotton is the first transgenic crop. Which gene was introduced in the first transgenic cow, Rosie? So the gene is human alpha lactalbumin gene has been introduced in the first transgenic cow. So in protoplast fusion, which chemical is used? So DMSO is dimethyl sulfoxide, which is the volatile gas from the onion. That is the reason that you cry whenever you cut the onion. Dimethyl sulfoxide. Liquid nitrogen, they are used for cryo preservation. Pectinase, it's an enzyme which can break down pectin. PEG, PEG is nothing but polyethylene glycol. This is used even in hybridoma technology when we fuse the myeloma cells and the antibody cells, antibody producing cells, and we get this hybridoma cells. So even there the chemical fusion can be brought about by polyethylene glycol, what we call it as PEG. Chemofusion and electrofusion are employed in protoplast fusion. Again, these chemofusion PEG, they have not informed in our NCERT book, but you should be aware about it. So in chemical fusion, PEG can be utilized. Electrofusion, in high electric fields, two protoplasts of different species can be fused, and we call this as protoplast fusion. Even this high electric field can be used even for hybridoma technology. From hybridoma cells, we get monoclonal antibodies. So that is again outside of the syllabus, but it's a part of the biotechnology processes. A functional adenosine deaminase cDNA can be introduced into cells of the patients receiving gene therapy by using vector constituted by. So for most of the animal experiments, the gene therapy or the transfer of our DNA, we go for this disarmed pathogens like retrovirus. So they just introduce the gene of interest into the human beings without causing any diseases because they have been disarmed. So a functional ADA cDNA can be introduced in cells of patients receiving gene therapy through retrovirus. Bt cotton is not a genetically modified plant, insect resistant, bacterial gene expressing system, or resistant to all pesticides. So the 10th one, the incorrect statement is resistant to all pesticides. They are resistant to, the Bt cotton is resistant to only to the insect, that is bollworms, okay? But they are not resistant to pesticides. Pesticides are chemicals which are sprayed on plants to prevent from the insects growing on them. So they are insecticides which we utilize. So but Bt cotton is not resistant to all the pesticides, that we have to remember about. A bioreactor is a fermentation tank, culture of bacteria, hybridoma, culture for synthesis of new chemicals. So a bioreactor is a fermentation tank where we culture the transformed cells and obtain the desired recombinant proteins. 12th question, a probe, which is a molecule used to locate specific sequences in a mixture of DNA or RNA molecules, can be, usually they are radioactive nucleotides will be there. So the probe. So what is the probe molecule made up of? Is it made up of a single stranded RNA or a single stranded DNA? Either RNA or DNA. Can be single stranded DNA but not single stranded RNA. The answer is the probe can be either RNA or DNA, but they should be radioactive nucleotides, so that by the emissions of radiation and auto radiography we can identify where they have tagged with its complementary DNA or RNA. That is the purpose of them being radioactive nucleotides. Anyway, 13th question, C-peptide of human insulin is a part of mature insulin molecule. In mature insulin molecule they have only A and B chain. So responsible for formation of disulfide bridges, they are not responsible for it. Removed during maturation of proinsulin to insulin, this is the correct answer. Removed during maturation of proinsulin to insulin. In mammals this is happening. As per our textbook they have given it as mammals. Even in human beings the C chain is removed and proinsulin is now, after the removal of C chain, proinsulin is activated as insulin, which is an active hormone. So 13th one, removed during, C peptide is removed during maturation of proinsulin to insulin. The 14th question is the trigger for activation of toxin of Bacillus thuringiensis is. So it is nothing but the alkaline pH of gut. The alkaline nature of gut is the triggering mechanism for the activation of toxin, which is in the form of protoxin when it is secreted from Bacillus thuringiensis. Why they are secreted as protoxin within the bacteria is to prevent the digestion of the bacteria which has secreted it. They will be released as protoxins. So the alkaline pH of gut, they are going to be activated as toxin. The production of value-added products like nutrition supplements, pharmaceuticals, fuels, etc. using transgenic crop is called as, so since here we are obtaining molecules, so it can be called as molecular farming. So we're using biotechnology products, we can, or transgenic crop, the molecular farming can be done, like we can obtain nutritional supplements, pharmaceuticals like antibiotics, vitamins, enzymes, fuels, etc. can be obtained from transgenic crop. That is the reason the appropriate answer is molecular farming. Which Indian plants have been either have been patented or attempts have been made to patent them by Western nations for their use? Basmati rice, turmeric, neem, all of this they have tried to patent, even though it's a traditional knowledge and prevalent in India from long. So all of this is the answer. Biopatent means right to use an invention. Biopatent is majorly meant to protect the intellectual property rights of companies. And here it is, since it is biopatents, it is concerned with biological inventions that they have made. Right to use biological resources, that cannot be, it's a general answer. Anyone can use biological resources. For that, biopatents is not required. So whereas the biopatent rightly implies right to use an invention. So modified biological resources or invented biological resources, then we could have utilized it as an answer, but it is not completely right. So the right answer would be right to use an invention. So again, there is a little ambiguity. Right to use an invented biological resource would be better. Anyway, so since they have used biopatenting resources, here ambiguity is there. So we'll stick on to right to use biological resources or modified biological resources should be there. So that would be appropriate answer. So we can avoid this right to use an invention. I feel that right to use modified biological resources would be, then there wouldn't have been an ambiguity in the question. Anyway, so in the final exams, they would be giving the appropriate answer. A transgenic food crop which may help in solving the problem of night blindness in developing countries is golden rice, because the purpose of producing this golden rice was to see to it that the people Ingopotricaceae developed golden rice to overcome the vitamin A deficiency, or VAD, what we call it as. So a transgenic food crop which may help in solving the problem of night blindness in developing countries is golden yellow rice. Which of the following statement is false? Insulin was originally extracted from pancreas of slaughtered pigs and cattle. Animal insulin is difficult to obtain. Animal insulin is identical to human insulin. Non-human insulin causes some patients to develop allergy. So most of your examinations, you can expect incorrect statement or find the odd one out, because the examiners, paper setters, they want you to utilize more time to read the question. So they want you to spend at least 45 to 50 seconds before you complete the answering. So the answer for this is, the incorrect statement is animal insulin is identical to human insulin is an incorrect statement. Which of the following is correct? The proteins encoded by the genes Cry1AC and Cry2AB control cotton bollworms. Protein encoded by Cry1AB controls corn borer. Proteins encoded by Cry1AC and Cry1AB control flies. So here the options A and B are correct. So this has been given, isn't it? For corn borer it is Cry1AB. For cotton bollworms it is Cry1AC and Cry2AB. Why insulin not administered orally to diabetic patient? The reason will, insulin will, here with the options that they have given, so insulin will lead to a sudden drop in blood sugar if given orally. And another thing is insulins are made up of proteins and they are digested within our digestive system. So that also we should be remembering about. If they give that option, that is a correct one. So insulin will lead to sudden decrease in blood sugar if given orally. Apart from this, insulin will also be digested by the gastric enzymes within the stomach itself. They would be digested by the proteinaceous enzymes. Okay. Consider the following statement. Bt toxin gene has been cloned from bacteria. Genetic engineering works only on animals and has not yet been successfully used on plants. Strains of Bacillus thuringiensis is used as bioinsecticidal chemicals in plants, or bioinsecticidal in plants strains. So which of the statement given above are correct? So this is what the options they have given. For this, the answer would be 1 and 3. So Bt toxin has been cloned from the bacteria, that is gene has been cloned from bacteria, yes, that is appropriate. Genetic engineering works only on animals and has not yet been, that is a wrong statement. Third one, strains of Bacillus thuringiensis are used as bioinsecticidal in plants, that is correct. They are used for controlling the bollworms, corn borers and others. So first and third statement are correct. This is what is the answer for it, which is not true with respect to transgenic animals and their contribution to human welfare. Transgenic mice are being tested to ensure safety of polio vaccine. Rose's milk contain human gene insulin. This itself is wrong. The transgenic cow contained human alpha-1 lactalbumin protein. They did not contain human gene insulin. Transgenic cows produce milk with less fat. Transgenic sheep grow more wool. So these are all correct statements. Rose's milk contained human gene insulin is a wrong statement. It is very noticeable. When DNA is transcribed into mRNA, usually the mRNA remains single-stranded, but in some cases an RNA can be made that is complementary to the mRNA. This is called dash and its main function is to dash. So the 24th question, the answer is antisense RNA. So the mRNA remains but in complementary to the mRNA. So we call it as antisense RNA. It blocks gene expression. So double-stranded RNA, because they are complementary sense and antisense, they are going to form double-stranded RNA and they will be blocking gene expression, which is RNA interference, where the double-stranded DNA breaks the mRNA into fragments. That you should be aware about. A patent is granted for an invention, an improvement in earlier invention, process of generating products. Fourth one, strains of microorganism. So that we should be knowing about. So the patent is granted for which of it? So the 1, 2, 3, not for strains of microorganism, an invention, an improvement in an earlier invention, process of generating a product, these are all provided patents. So the answer is 1 and 3. 1, 2 and 3 is the correct answer. With this, I'll be completing this session of the multiple choice question training for the biotechnology applications part of it. Thank you for attending the class proactively, and hope you all get into very good medical colleges by very good performance in botany and other subjects too. Thank you all. God bless you all. Bye.